5 Landmark Events That Changed Hospital Pharmacy, Part 2. Hi again, everybody, and welcome back. The last time we met, we talked about how medication errors and unsterile compounding can negatively impact patient lives, and as a result, how pharmacy faced greater oversight and scrutiny by regulatory agencies to reduce the problem. Now we will move on to discuss more positive and promising events that relate to cancer therapy, as we go over Landmark Events 4 and 5. Event number 4, the discovery of imatinib. Imatinib, also known as Gleevec, was called a magic bullet when it revolutionized the treatment of cancer, specifically chronic myeloid leukemia, CML, in 2001. Imatinib was invented by biochemist Nicholas Lydon, then working for Ciba-Geigy, now Novartis. The use of the drug to treat CML was driven by Brian Drucker, an oncologist at the Dana-Farber Institute. Discovery of the Philadelphia chromosome in leukemia. In 1960, Drs. Knoll and Hungerford were examining cancer cells from 2 patients with CML and noticed something peculiar. One of the 46 chromosomes was abnormally short. They examined leukemia cells from 7 more patients with CML and found they also had the same abnormal chromosome. This tiny chromosome was later named the Philadelphia chromosome, after the city where Drs. Knoll and Hungerford discovered it. Further research showed that 95% of patients with CML have the Philadelphia chromosome. When techniques for studying DNA improved in the 1970s, researchers realized the Philadelphia chromosome forms when 2 chromosomes break and the pieces swap places. A few years later, Nora Heisterkamp, MD, and her colleagues discovered that when the Philadelphia chromosome forms, 2 genes that are normally separated get fused together. The hybrid, called the fusion gene, is known as BCR-ABL. This is illustrated in the diagram to the right. Owen Witt, MD, and his colleagues at the University of California, Los Angeles, later proved that when BCR-ABL forms in blood cells, it causes CML. Here are the normal chromosomes 9 and 22. The chromosomes will each individually break at certain points, forming changed chromosome number 9 and also changed chromosome 22, which is the Philadelphia chromosome, the BCR-ABL. CML is caused by one translocation that creates a singular mutation, the BCR-ABL fusion gene, or the Philadelphia chromosome. Enter Dr. Brian Drucker, who guided the discovery and testing of imatinib. Dr. Brian Drucker, an oncologist at the Dana-Farber Institute, worked with Dr. Nicholas Lydon, a biochemist who led a drug discovery group at Ciba-Geigy, now Novartis. The pair searched through an extensive collection of compounds for one that could block the activity of the BCR-ABL fusion protein. They identified a drug that was incredibly good at killing CML cells. The drug was named STI-571 and later renamed imatinib or Gleevec. Imatinib is a tyrosine kinase inhibitor. In 1998, Dr. Drucker and his colleagues tested imatinib in a phase 1 clinical trial partially funded by the National Cancer Institute. The drug caused cancer to disappear in the majority of patients with CML that was in the early or chronic phase of the disease. 5 years later, 98% of patients from this trial were still in remission. Subsequent clinical trials found similar results in larger groups of patients. Compared with the standard treatments for CML, imatinib also improved patients' quality of life and caused fewer side effects. In 2001, the Food and Drug Administration approved imatinib to treat patients with CML that have the Philadelphia chromosome. Today, someone with CML who is in remission after 2 years of imatinib treatment have the same life expectancy as someone who doesn't have cancer. In addition to transforming the outcome for patients with CML, the discovery of imatinib helped establish a group of new cancer drugs called targeted therapies that are designed to attack cancer cells with specific genetic abnormalities. Targeted therapies have immensely changed how cancer is studied and treated, and has fostered a precision medicine approach, treatment tailored to the unique genetic changes in an individual's cancer cells. Imatinib turned off the light switch and only killed the cancer cells, reducing significant side effects. These targeted therapies have immensely changed how cancer is studied and treated. Event number 5, cancer immunotherapy. What is cancer immunotherapy? With cancer immunotherapy, we are using the patient's own immune system to fight the cancer. We can administer immune system components to the patient, such as man-made proteins, or we can stimulate the patient's own immune system by utilizing T cells to attack the cancer cells. We boost the body's immune system or train the immune system to specifically attack some part of the cancer cells. These efforts are a departure from most traditional cancer treatments, such as chemotherapy and radiation, which take direct aim at the tumor, generally to only limited effect in patients with advanced cancer. Here's an illustration comparing the traditional cancer therapies to cancer immunotherapies. With traditional cancer therapies, radiation or drugs will kill not only the cancer cells, but also kills healthy cells, causing many side effects. Whereas cancer immunotherapies, the immunotherapy unleashes the patient's own immune system that kills the cancerous cells selectively, with few to no side effects. Bacteria and viruses have proteins on their outer surface that are not normally found in the body. Our immune system sees these proteins as antigens and attacks them. Cancer cells are different from normal cells in the body and have clear differences. Our immune system doesn't always recognize cancer cells as foreign, or the response might be only modest. T cells are the warriors of the immune system, responsible for hunting down and destroying infected or abnormal cells. For years, scientists thought that our immune system didn't recognize cancer tumors at all. Then research on biopsy specimens revealed that T cells often succeed in infiltrating the environment around tumors, but they either fail to mount an adequate response or hold the cancer at bay before finally being overmatched. This is what we would like to see with cancer immunotherapy: a T cell approaching a cancer cell, the T cell attacking the cancer cell, and the cancer cell being destroyed. When the T cell does recognize the tumor, it amounts to an ignition switch for the immune response. But just turning the key isn't enough. T cells generally need a second signal to become activated against the cancer, a so-called gas pedal. Then finally, the brakes must be released. Here's an example. Initially, the antigen on the cancer cell attracts the T cell. A second interaction called activation then occurs, where molecules from the T cell interact with the cancer cell. The immune system then applies its brakes to prevent it from going out of control. In this case, it is CTLA which prevents the T cell from secondary activation. To override the CTLA, drugs such as Yervoy blocks the CTLA brakes, allowing activation of the T cell. The activated T cell can now attack and destroy the cancer cell. Checkpoint inhibitors. The immune system has the ability to keep itself from attacking normal cells in the body. To do this, it uses checkpoints, molecules on immune cells that are activated to stop an immune response, the so-called brakes. Cancer cells take advantage of these checkpoints to avoid being attacked by the immune system by making sure the brakes are set. Checkpoint inhibitors are monoclonal antibodies that work by releasing the natural brakes on the immune system, enabling T cells to attack tumors. Here's another diagram illustrating the effect of checkpoint inhibitors. First, the T cell undergoes primary activation with the antigen of the cancer cell. The T cell then applies its brakes or immune checkpoint to prevent it from going out of control. With the use of checkpoint inhibitors, these brakes can be released to allow the T cell to destroy the cancer cell. The first immune checkpoint receptor that was discovered was CTLA4. CTLA4 is a checkpoint molecule protein found on the surface of T cells. CTLA4 modulates the early activation of systemic T lymphocyte immunity. It normally acts as a type of off switch or brake that helps keep the T cells in check by preventing activation, preventing them from attacking normal cells in the body. Ipilimumab, or Yervoy, was the first checkpoint inhibitor approved by the FDA in 2011. It is a monoclonal antibody that attaches to the CTLA4 receptor and stops it from working. This boosts the immune response against cancer cells in the body. Checkpoint inhibitors, PD-1. PD-1 is another checkpoint molecule protein on the surface of some T cells where it helps keep the immune response under control. It does this when it attaches to PD-L1, a protein found on some normal and cancer cells. When PD-1 binds to PD-L1, it basically tells the T cell to leave the other cells alone. PD-1 seems to be found more often in T cells in tumors and inflamed tissues. Cancer immunotherapy drugs can target either PD-1 or PD-L1, thus boosting the immune response against cancer. PD-1 interacts directly with cancer cells, so these drugs may be more potent and less toxic than chemotherapy. Some cancer cells have large amounts of PD-L1 on their surfaces, which help them evade immune attack. So in certain cancer types, such as non-small cell lung carcinoma, bladder cancer, and melanoma, the PD-L1 biomarker has identified patients with a higher likelihood of treatment response. Here again is another diagram showing how the PD-1 checkpoint prevents activation of the T cell, and how the use of checkpoint inhibitors release the brakes and allow immune attack of the cancer cell by the T cell. Some examples of PD-1 checkpoint inhibitors are nivolumab, Opdivo, which is manufactured by Bristol Myers Squibb, and pembrolizumab, or Keytruda, manufactured by Merck. Cancer immunotherapy is still in its early stages, and more consistent, rigorous studies are required. Immunotherapy drugs are now affecting tumors in certain patients, unlike almost anything seen with conventional treatments. However, the promise of advancement can be thwarted by the cancer's ability to develop resistance to medicine's attacks, as well as its extremely high cost. Still, there is no doubt that cancer immunotherapy has revolutionized cancer therapy. Compared to the previous landmark events, Events 4 and 5, the discovery of imatinib and cancer immunotherapy, both deserve bright stars for a promising future. We've got a lot in store at the PharmEasy Tutor channel. There will be upcoming talks on anticoagulation, electrolyte management, quinolone side effects, and much more. So please stay tuned to us. Thanks for tuning in to watch this installment of the PharmEasy Tutor. I hope you learned something that you could use at school or in practice. If you'd like to continue to see more of these types of tutorials on YouTube, please make sure to click on the subscribe button below to change it from red to gray. Also, if you like this video, I would appreciate it if you can click on the thumbs up icon below to change the color to blue. If you have any comments or questions, please feel free to add them in the comments section below, or share this site with someone else. Stay tuned to the PharmEasy Tutor channel for more lectures in the upcoming weeks. So until next time, remember to take it easy.